Happy daylight savings time. Yes, we're all here, we're all awake, we're all caffeinated. Good. Couple of things that I wanted to mention today, some of them based on George's lecture from Friday. He came up with this great discussion here, I think, think, talk, click. I'm actually going to change that ever so slightly based on what I heard from our seminar speaker on Thursday, and that's going to be click, think, talk, click. Yeah, it just rolls off the tongue, doesn't it? But the basic idea here is for everyone to click first before talking, and I suppose we could switch these two as well. But the idea here is that you have your own idea what you think about a clicker question, and then you can discuss it with your neighbor, anyone else in the class for that matter, and then change your vote afterwards if you think that your neighbor was right and you were wrong, or both of you are wrong or both of you are right, as the case may be. The other thing that I was highly recommended to do was make clicker questions way harder, which I'm not sure if is necessarily what people would like me to do, so we shall see. You can decide whether they're now harder or not, as the case may be, but it gives us more time to discuss them afterwards. The other highly recommended thing that Dr. Freeman suggested was, instead of asking for volunteers when I ask questions, is to go down a class list and pick names randomly. I'm probably not going to be able to do that this term, but everyone can stop signing up for virology, because I'm probably going to do it next term. Which is a reminder here for looking in the middle, where we have still a few spaces in both of the lab sections. This is the mutant viruses from Hell Lab. There are 3 spaces left in the Tuesday, Thursday, and 6 spaces left in the Monday, Wednesday lecture. There are still a few spaces left in virology, and there'll be a lot more because everybody's going to drop after I said I'm going to ask you questions randomly. And then there also are quite a few spaces left in the literature review, Chem Bio literature review, cross-listed in both biology and chemistry. I know a couple of you have asked me about this class already, and I've said you can sign up now because the people in the lab class have had their chance, and if they haven't taken it, that's their problem. So at this point, okay, go down my list. Any questions from? No. Any questions about this so far? Yes, no? Okay. So we shall now try, for the first time, for me anyway, to do this new clicker thing. So, plan with the new clickers, for those of you who got in here late, is we'll do a minute, everybody gets to score, then you can chat with your neighbors, and then you can try again. Sound like a good deal? I'm only going to count the second round. Good? Yes? No? Huh? We got it? We ready to go? The other thing that Dr. Freeman suggested is don't read the question, because I could have some, I don't know, something in my voice that says you should answer C. But I'm still going to go ahead and do it anyway, just in case there are people who have trouble reading or this kind of thing. So this is, again, minute but quiet minute. To generate a C-terminus of a protein with alternative sequence, which of the following methods of post-transcriptional regulation would be the most likely? Riboswitch, alternative splicing, RNA editing by ADAR, RNA export, mRNA localization. Okay, so that's your first minute. Now turn to your neighbor and tell them why you chose what you chose. We ready to click again? Yes? Okay, let's give it another try, starting from 0. And okay, was that better or worse? Better? We like more time to think? Actually didn't change results much at all, interestingly enough. I could see them up here. So C-terminus of a protein with alternative sequence. Why not riboswitch? Riboswitches mostly have to do with either turning on or turning off transcription. So that's not going to change your sequence of your protein. Alternative splicing, what's that going to do to the N-terminus of your protein? Keep it the same. What might it do to the C-terminus? Potentially change it. RNA editing by ADAR. What does ADAR usually do in terms of RNA editing? Changes one base. So if you're changing one base, what is that likely to do in terms of the rest of your protein? Maybe it'll give you a stop codon. That's distinctly possible. It could also change one nucleotide and maybe one codon. Is that going to change a lot of a particular C-terminus of a protein? Could be the N-terminus, could be pretty much anywhere. RNA export, probably not going to make a big difference. mRNA localization, not as well, and nobody really liked those. So what does that leave us with? Alternative splicing. So the other thing with this whole business of checking out how these things work better or worse, we get to experiment. And what do us scientists like to do? We like to experiment. So unfortunately, I should have done IRB. You've heard about IRBs? Institutional review boards. If I use you for scientific experiments, I actually have to get it cleared by somebody else as well as you. But only if I use it for somebody other than me in terms of the results. Okay, so today we're going to finish up talking about translational control and how literally translation is regulated. And again, this is something we could spend a whole course talking about. And then towards the end, I want to start talking a little bit about methods. And one of the things I'd really like to get your feedback on, we will have online evaluations. But one of the things I'd really like to get some feedback on is whether you think we should do some of these method things at the beginning of the course rather than right now at the end of it. So, but first, a little bit about translational control. If we think about translational control, there are sort of two major ways that we can regulate translation. This has to do with the two different kinds of translation that we have. We've got bacterial-like translational regulation and eukaryotic-like translational regulation. Bacteria are dependent on what? Shine-dalgarno, ribosome binding sites, whereas eukaryotes are much more dependent on caps. and interactions with the cap and the tail structure. So if you think about translational control in bacteria, very often this has to do with just changing access to the Shine-dalgarno sequence. Interestingly enough, access to AUGs in eukaryotic systems are also pretty similar. So if you have some kind of secondary structure or a particular protein that's going to associate with it, if it interacts with the Shine-dalgarno sequence or blocks the AUG, you're not going to have translational regulation. Often you have these ribo switches, and here this is a ribo switch which is just controlling whether you have translational initiation or not. So it's a secondary structure which forms in the RNA, and of course this forms a tertiary structure that now interacts with something. These RNAs that interact with some kind of small molecule are called aptamers. And if they interact with a small molecule, it changes that secondary structure, and here that change in secondary structure blocks the binding site for the ribosome. One of my favorites in terms of sort of the simplest ever kind of regulation you can get in terms of translation are what are called RNA thermometers. These RNA thermometers are shown up here in the upper right, and it's literally a secondary structure that forms in the RNA that just by changing the temperature, that secondary structure changes. And so just by heating up the cell, say like a heat shock, and this would be a nice way to regulate some of those chaperones, the heat shock proteins, for instance. And in fact, this is how some bacteria regulate their heat shock proteins, is just by changing the temperature, you change the secondary structure. Now you have access to this ribosome binding site. And then the final one, which is a nice connection back to George's lecture from Friday, is thinking about small RNAs. And so George talked about the small microRNAs, the siRNAs, the long non-coding RNAs, and of course the CRISPR RNAs. Oh, and by the way, he will be writing a couple of questions for the exam, and I will be writing a couple of questions for the exam, which is when? Next Tuesday. What time of day? Daylight savings time. So ridiculously early in the morning. So yeah, I'll be writing a couple of questions on that as well. But these are short RNAs which literally all they do is bind to messenger RNAs through base pairing interactions and just block the normal binding, for instance, you would have with the Shine-dalgarno sequence. So these are some really RNA-based mechanisms. There are also some protein-based mechanisms in terms of regulating translation. These have to do with all of those initiation proteins that, of course, all of you remember from the last midterm. Particularly eukaryotic initiation factor 2. What does eukaryotic initiation factor 2 do? Again, I should be going down my list of the class and picking out a name. Since I don't have my class list with me, I guess I won't. So EIF2, eukaryotic initiation factor 2, interacts with the tRNA that you're going to need to start translation with. And IF2 is the same way. It's a GTPase, binds to GTP. When you have the appropriate interactions, GTP gets hydrolyzed to GDP, and the RNA is released. You assemble the ribosome, and you get translation. What happens to EIF2 after you have GTP hydrolysis? You now have GDP bound to your protein. In the case of most of these GTPases, what do you need after that? You need a nucleotide exchange factor that takes the GDP off and puts GTP in. That particular exchange factor is called EIF2B that stimulates this process. So this is what happens normally. No problem. Again, need lots of GTP for translation. However, under stressful conditions, when you have virus infection, EIF2 gets phosphorylated by specific kinases. What happens when it's phosphorylated? When it's phosphorylated, it sticks to EIF2B and can't bind to GTP anymore. What does that mean about translation, and particularly translational initiation? You don't get any. Exactly. So it stops translational initiation. And this could make sense if you're under a stressful condition. So we've talked about before, translation takes huge amounts of energy, so you're going to save yourself some energy by not undergoing translation. And if you've just been infected by a virus, the virus is trying to convince your translation system to make more virus rather than make more cell. So again, it might be a good idea to slow down this kind of translation process. And even in cell cycling, in most of our cells, they're in a quiescent phase, the gap 0 phase. Turns out that the gap 0 phase is also regulated by this phosphorylation and slowing down translational initiation. So that's a protein-based way now that we can slow down translation. But viruses, of course, are ridiculously smart, so they've come up with other ways of dealing with this issue of translational initiation. One of these is what's called an IRIS, or internal ribosome entry site. Internal ribosome entry site. And basically what the IRIS is a sequence which forms a structure in your RNA. And that structure can now associate with the other proteins that are required, and particularly the proteins that are required for getting translational initiation in a cap-independent fashion. So this secondary structure here, in this particular case, turns out there are multiple different irises, originally found in viruses, where you have interaction with a poly-A tail and this iris, but no cap structure, which is here, and you still get translational initiation. Normally, again, you'd have a 5' cap and your cap binding protein. This would provide the signal for translational initiation, but these irises provide another way of doing that. So this is just translational initiation. Another way of regulating the amount of protein you get is the stability of your messenger RNA. Most bacterial messenger RNAs are really not very stable at all. They're made, and since we have co-transcriptional translation, they're also being translated, but they're also being degraded really quite quickly. So you have a lot of turnover, as we call it, in terms of your RNAs. On the other hand, messenger RNAs that a eukaryotic cell has spent all of this time splicing, capping, tailing, etc., those last considerably longer, anywhere from half an hour to tens of hours. And this is the, this allows you to be able to translate these messenger RNAs multiple times, usually the polysome, those ribosomes which are going around and around. So how do you sometimes stop this kinds of translation? Well, usually what happens with a standard eukaryotic messenger RNA, oh, sorry, it should be over here, is you have your cap, all of your exons spliced together, and a tail. There are a couple of things to point out here. We've talked a little bit about these UTRs before. What does UTR stand for? Exactly, untranslated region. So we've got 3' UTRs and also usually 5' UTRs, which are present in pretty much all eukaryotic messenger RNAs. In a normal case, you have this poly-A tail, again, a couple hundred nucleotides at the end. This slowly gets chewed up by 3' to 5' exonucleases. Why do we have the poly-A tail? It seems to mostly be protecting the rest of your coding messenger RNA. Once this poly-A tail gets to a really short length, and this is probably because you don't have much binding by poly-A binding proteins anymore, which also help to stabilize that poly-A tail, then the whole rest of the messenger RNA gets chopped up really quickly. And most of that happens due to endonucleases that happen in the middle of your messenger RNA. Once you chew up to here, then the rest of it gets chewed up, and this gets chewed up very rapidly, partly because the cap is removed. Now, in some cases, if you have a messenger RNA that you want to have be very unstable, i.e. you don't want to make very much protein from it, often these will have specific endonucleolytic cleavage sites right in the middle of that RNA, and usually in these UTRs. If now you have the activity of this endonuclease, it'll cut in your messenger RNA. Now you don't have a poly-A tail to protect you anymore, so you end up at this step already. and your messenger RNA gets degraded really rapidly. It's just another way of looking at that. Here we have our normal messenger RNA, perfectly happy. As soon as you start to degrade this beyond the point that you can have a poly-A binding protein associated with it, then the rest of your RNA is going to get degraded really rapidly. So this gives you an idea how long you're likely to have a messenger RNA present inside the cell. We know about this in terms of some specific regulation. The best understood of these is iron regulation. Iron is a very interesting atom in biology. It's highly reactive and causes something called the Fenton reaction, which causes a lot of DNA damage, but it's also absolutely required in a number of enzymes in terms of their activity. So the exact amount of iron present inside a cell is really important. And the way that the cell deals with this is it has two, well, there are more than this, but two main regulatory proteins. There's this protein called transferrin receptor. The transferrin receptor brings iron inside the cell. So if you have no iron, you want to be sure to be bringing iron inside the cell. But if you have too much iron inside the cell, you have this protein called ferritin, and ferritin makes this cage and basically protects the rest of the cell from your iron. So this is regulated based on mRNA stability with one particular protein. It's called an aconitase. Here we're just thinking of it as an iron-binding protein. It also has some enzymatic activity that, if people are interested in, we can talk about later. So just basically an iron-binding protein. This iron-binding protein has iron as an allosteric regulator. So this allosteric regulator, once it binds to iron, will come off of your RNA. Same protein regulating these two different genes. Ferritin, again, when you need to have this, you've got too much iron around. Too much iron around, you're not going to bind to the messenger RNA. You're not going to block the ATG. Too much iron around, you will release this stabilization in the 3' UTR, which then causes this messenger RNA to get degraded very rapidly. Same protein, two different genes which it's regulating through this messenger RNA stability. Well, actually, in one case, it's actually just the translational initiation. So translational initiation of ferritin in the absence of one of these molecules, and destabilization of the messenger RNA for the transferrin protein. Now, what kinds of proteins do these sort of look like? Binding to a ligand, changing nucleic acid binding. Where else have we seen this? Again, where's my class list? I could go down and find somebody. So lactose in terms of the lac repressor binding to a small ligand, causing changes in terms of the structure, in terms of the lac repressor, binding by lactose, that comes off of the DNA. So it would be a very similar kind of process. So it's an allosteric change in nucleic acid binding. And you know Stedman loves these kinds of questions on exams, right? Speaking of questions, so we're going to do click or think, click, no talking, talk, click again. Right? Ready to go? Good. Okay. So I'm going to start this. This would help. And yes, I will read these. Majority of mRNA degradation takes place by 5' to 3' endonucleases, 3' to 5' endonucleases, 5' to 3' exonucleases, 3' to 5' exonucleases in the nucleus. And you've got a minute to make your first decision, and then you'll have a minute to chat with your neighbors about why you made that decision. Okay, we're at this stage. Tell your neighbor why you made that decision. Okay, we ready to go again? Yes? Okay, let's go. Seven, 5. This one actually was a considerable change between the two rounds. A lot more people liked B the first time around, and more people liked D this time around. And I actually agree with you. So that's a good thing. So why did nobody like E? I know, because I can't ask you why nobody liked E. Well, you should have told your neighbor why E was a really bad idea. So E is wrong because why? Where are messenger RNAs? Where are they being degraded? Not in the nucleus. They're being degraded in the cytoplasm. So the big difference here seems to be between endonucleases and exonucleases. What's completely wrong about A and B? Again, I need my class list to find somebody on this. But what about endonucleases? Do endonucleases have a 5 to 3 or a 3 to 5? No. An endonuclease just cuts, and I should have brought my model, right in the middle of your DNA. So there's not a 5' to 3' endonuclease or a 3' to 5' endonuclease. So that couldn't be right. Okay, so that leaves us with 5' to 3' and 3' to 5' exonucleases. What's mostly being chewed in a messenger RNA? The poly A tail, exactly. And poly A tail is at what end? The 3' end. So if you're chewing into the 3' end, you're going from 3' to 5'. Okay, we happy? Yes, no? Make more sense? Not? Good to do this round? More time? Yes, no? Huh? Whatever, just move on. Okay. Okay, so that was it for post-transcriptional control. Let's talk methods. And again, please let me know if it's something that you think I should have talked about way earlier on in the course. Today we'll talk about proteins and cells. On Wednesday, we'll talk about nucleic acids. And on Friday, we'll review everything that we've talked about so far. And on Tuesday, we'll have an exam. Where did this quarter go? I don't know. So why should we care? Basically, everything that I've talked about and we've discussed so far depends on what we're going to talk about in the next 2 lectures, or the end of this lecture and the next lecture. So I think it's really pretty critical. One thing that I need to do more of is something that I think George did a really nice job with, which is having some outlines and going back and recovering some of those outlines. Revise, review, all those kinds of things. Haven't done a great job of that. But in terms of overall conceptual ideas here, I want to start talking about centrifugation, and then we'll talk a little bit about chromatography. We talked about chromatography briefly already. SDS page, I actually changed the order here a little bit. Mass spectrometry. Mass spectrometry turns out to be basically the way that people do proteomics these days. Large structural measurements of proteins, and then individual proteins, some structural methods, and if we have a chance, we'll talk about bioinformatics, but it'll probably be on Friday, tomorrow, on Wednesday. One of these days, anyway. So today, mostly going to talk about SDS page and centrifugation, a little bit of chromatography as well in the next 20 minutes or so. SDS page, what does SDS page stand for? Sodium dodecyl sulfate polyacrylamide gel electrophoresis. All of you read about it in your textbook, right, before class? It's another thing that I'm supposed to do and get people to do before they come to class is do the reading. We shall see. Centrifugation, there are basically 3 flavors of centrifugation: preparative centrifugation and analytical centrifugation. Analytical centrifugation is both sedimentation. You remember those crazy S numbers we were talking about when we talked about ribosomes? Where do those S numbers come from? Sedimentation, analytical, ultracentrifugation. And then equilibrium. When do we talk about equilibrium ultracentrifugation? Okay, where's my class list? Okay, we'll find it. Michelson and Stahl. What were Michelson and Stahl doing? DNA replication. So this was equilibrium gradient ultracentrifugation. Chromatography, talked about chromatography already in terms of what? Again, where's my class list? I need to find some of these things. Isolation of what? Binding proteins, DNA binding proteins in particular, DNA affinity chromatography. Great review for something that's coming up next Tuesday. Today we'll talk about some less specific methods, ion exchange and gel filtration, and of course this affinity chromatography. And if we get a chance, we'll talk a little bit about 2D gels and mass spectrometry, and then some of the structural techniques for trying to determine what protein structures look like, X-ray crystallography and NMR spectroscopy, although I suspect we're not going to get to those. So SDS page is a really nice example of what we call gel electrophoresis. And gel electrophoresis takes advantage of sizes and charges of macromolecules. And today we're going to be talking about proteins, but on Wednesday we'll talk about nucleic acids basically in the same way. So gel electrophoresis, you have a electric field set up from a cathode to an anode. The way I remember cathodes and anodes is cats like to take things apart. Anyone's got a kitten or something like that knows they're really good at shredding things. So that's your negative going to the anode, which are your anabolic steroids for building things up. So that's the way I remember. Other people have other ways of remembering these things. So negative going to positive. So these are going to separate, again, based on charge, because that's your negative going to positive. What's in between is the gel. And so your gel electrophoresis is basically a whole set of holes. And those are just going to slow down the progress of whatever you're trying to get to move in this electrical field. So bigger stuff is slower, smaller stuff goes faster. Make sense? Okay, so one of the big problems, of course, is that if you're talking about proteins, unlike nucleic acids, proteins have all kinds of different charges on them. And they're going to have lots of different sizes, and also the shape that that protein has is really going to make a big difference in terms of how it's going to go through one of these gels. So how do we deal with this? The main way we deal with it is to, as always in good molecular biologists, blast everything apart, right? And try and simplify it as much as possible. So the way that that happens is we use detergents, and one of the favorite detergents is something called sodium dodecyl sulfate. Why? Because it has a nice charge at one end. This is our sulfate. This dodecyl group here is clearly highly hydrophobic. So what do? You take your protein, you add this detergent, and you heat it up, usually boil it for a couple of minutes. And what that does is it allows the detergent to come into your protein and basically get rid of any of the structure, particularly the tertiary structure that you had in your protein, which means that now all you're going to be separating on is based on size. The other thing that it does is gives you a uniform charge because of the charge which is present at the end of this SDS. The other thing that's almost always used, although we'll see some examples of it when it isn't, is something which will break disulfide bonds. So we talked really briefly about disulfide bonds. We talked about protein structure again way back at the beginning. And including one of these disulfide breakers will pull apart any of these disulfide bonds, which would change the structure a little bit. Yeah, Diana?
Do you make a detergent specific to one type of protein, or is the detergent target a wide range of?
Okay, so the question is whether you have specific detergents for specific proteins, or do you try and have one which is really general? And sorry, I'm just paraphrasing the question. But the idea here is SDS is really very general. So it should work with pretty much all proteins. And usually, and as we'll see here, you're not usually just looking at one protein here. You're looking at a whole mixture of proteins. And so you want to just use one particular detergent. People use different detergents, but SDSs seem to work really well. So generally, people just use that one detergent. So let's look at an example of this. We'll look at some real SDS page gels a little bit later on. Here is your, okay, let's start at the top. Process, so again, we've got our protein here. It's got nice tertiary structure and also has a disulfide bond. You heat it up with this detergent, SDS, and a disulfide bond breaking reagent. Now we have these proteins that are unfolded and have these negative charges. You put them on a gel, you run them for a particular period of time, and you have these three bands which are present here. Now, how do you see these? How do you see proteins? UV light works well if you have something which is going to either absorb in the UV, or you have something which fluoresces after you shine UV light on it. Most proteins don't actually have too many of those. Use dyes, or down here at the bottom, stains. Now, what stains do you use? What kind of dyes? Actually, things people use for things like, shirts, fabrics, etc. Any kind of dye which has been developed literally for hundreds of years that will bind proteins. Classic one is something called kumasi, but it's originally developed as a fabric dye, which will bind to proteins. And so you're really looking at the dye here, which is now bound to each of these individual proteins. So you need to have a stain of some kind to be able to actually visualize these particular proteins. So here we've got three different polypeptides. This one is a protein with these two pieces attached by a disulfide bond. Here's a single subunit. And the nice thing about SDS page is now we're just looking at relative molecular masses. The B chain is bigger than the C chain, which is bigger than the A chain. And you can also do an experiment where now you don't treat with this disulfide bond breaking reagent, and you'd see what these two together would be even higher on the gel. They'd be at a different size. So SDS page, anyone who has worked in a lab or in some of our labs here at Portland State, I think in the cell biology lab, do you do it in principles? I know in the cell biology lab we do SDS page, and I know that in the biochemistry lab SDS page. This is a very, very common technique that is used throughout biology, and we'll see some of these things later on. Another technique which is used, oh, sorry.
I'm just kind of curious, is the only factor that leads to differences in how far the proteins move on a gel just the weight? Is it not have anything to do with like folded structure?
So the question is this, and again, I'm just paraphrasing here, correct me if I'm not getting your question correctly, but is it just based on relative molecular mass, or is there also some structural component? Because as I mentioned, these gels just have holes in them, and depending on how big that is. The idea is, and in fact, getting back to Diana's question is, do you use different detergents for this? So you make the assumption that the treatment of detergent is going to be elongating everything to basically your primary structure. And as it turns out, that's not actually completely true. So these are approximate molecular masses that you get in this process with the SDS page, and particularly at smaller proteins, you can end up with some pretty major differences. So it works reasonably well, but not completely. Yeah?
Is there ladders that are used to know what size, and are those specific to the protein you're working with, or structure?
Okay, so the question is, are there ladders that you can use for this? So what the heck is a ladder? This is what you buy in the little tube. No, these are molecular weight standards. And so you can get those, and those are usually set up in a way, again, with relative molecular masses, and those will have a particular distance that they will move through this gel, which is already known, and so you can say, okay, I've got this one, this corresponds to about 100,000, et cetera. But yeah, these are your standards that you would almost always use, and we'll see these when we look at our example in just a couple minutes now. Other SDS page questions? The wrong way. Try going this way. Another technique which is extremely commonly used anytime you walk past a molecular biology lab, you hear bzzz going on. That's because people are using centrifugation, and centrifugation as a way to separate different subcellular fractions or proteins from your vesicles, etc. And so this is just a cartoon of how that works. You start out by blasting your cell into lots of different pieces. Then you put it into a centrifuge, which will spin at various speeds. And we say a low-speed centrifugation here, it's about 1,000 times G. And what that will do is we'll separate most of your whole cells down here at the bottom from all of the rest of your proteins. And so this is what people would almost always call a crude extract, or just a supernatant, which has been added here. All the proteins that you're interested in are going to be up here. This is going to be the junk down here at the bottom. Almost always this gets thrown away. Then you'll take what's on top here, the stuff that's not gone down to the bottom of your tube, spin it again, and in this case you'll end up with most of your proteins up here in your supernatant, some of the smaller stuff, and the faster and faster you spin the centrifuge, you'll end up with the smaller and smaller things down at the bottom here. These centrifuges, particularly when you're doing very high-speed centrifugation is hundreds of thousands of times gravity. These are literally tanks. I had to move one a few months ago from one building to another. Nearly broke my toe in the process. These things are really, really heavy because it's one of the scariest things that you have in a lab. These things, if they're spinning at 100,000 RPM, piece of titanium that's about 30 pounds, it's really scary. And so they're armor plated around the outside. But these are great ways of separating different pieces, and particularly if you want to get ribosomes, viruses, etc., you do some slow spins to get rid of the junk, and then very high-speed spins to get these down at the bottom. So that's the preparative process, and almost everyone uses preparative centrifugation. Again, you've got your favorite protein. You want to separate it from all of the other proteins. Say it's a DNA binding protein that you think is going to be important for transcriptional regulation. You want to separate nuclei. You do a couple of spins, you get the nucleus, you purify the proteins just from the nucleus, because that's where you expect your DNA binding protein to be. So once you've done this purification process, now you want to analyze what you've got. And so there are two other ways you can do this with centrifugation. There's what we have on the left-hand side here. This is sedimentation, and this is equilibrium. So sedimentation, basically you have a sample that you put at the top of your centrifuge tube, and you spin it for a while. Now, spinning it for a while is usually tens of thousands of RPM for a couple of hours, and what happens is you'll have separation of big stuff, things like ribosomal subunits, from slightly less big stuff. And so whenever we talk about 30S, 70S, 16S, 18S, etc., all of this comes from these sedimentation velocity experiments. So you take your material, you put it at the top of your centrifuge tube, you spin it for a certain period of time, and what will happen is get separation of red and black, so slow stuff and fast stuff. The fast stuff is bigger than the slow stuff. And then you make a little hole in the bottom of your centrifuge tube and just start to collect what's in the bottom. So this is what's in the bottom, what's a little bit more than that, and each of these is going to be a fraction. So for instance, this bottom piece here ends up in this tube, the next piece ends up in this tube, then you end up with the black stuff you're looking for here in the next tube, and so on and so forth until you run through the end. So this is your fractionation process. Literally connecting stuff here starts at the bottom, moving its way back up to the top. Now, if you look for pieces of your ribosome, for instance, this would be a large subunit of your ribosome, the small subunit of your ribosome right here. So that's separating them. Yes?
Is that visible at the growth level? Or do you dye those as well?
Okay, so the question is, are these visible or do you end up dyeing them? This varies. And so in some cases, they actually will be visible, and you can literally see these actual things which are there. Usually you have some other way of detecting them. So it will be staining, and as we'll see in a couple minutes, there are other ways of trying to look at what's in each of those individual tubes. Usually, classic for molecular biology, it's a colorless liquid, and then you're like, okay, what the heck is in here? So there are other ways of trying to figure that out. Yes? So the question is how do you do this fractionation process? That's just paraphrasing your question. So literally, and you can come by my lab sometime and see that someone stuck a needle into the bottom of the tube and is literally just collecting drops as they come off the bottom. So you can literally count the number of drops that come off. You can do specific volumes for each of these, and fortunately people have designed machines that will also do this for you. Okay, so that's sedimentation. You can also do these equilibrium centrifugation experiments, and this was in fact what Mieselson came up with in the first place, which is probably why we always call it the Mieselson-Stahl experiment, why no one else had done it before, was to come up with both the math and the techniques for doing these equilibrium gradient centrifugations. These are much longer spins. This is up to 20, 30-hour spins that you do, because what happens is you set up a gradient, and that gradient has different densities. So the top of the gradient is low density, the bottom of the gradient is very high density. Has to do with the metal atoms which you put into that. And then you put in a secondary sample, and then what will happen is that secondary sample will end up going to where its density is. So you have a mixture of a sample, lots of different densities. As you spin it, the equilibrium shows up at one particular density. And very often you can actually see these. We do this with viruses all the time. And you can literally see a band where you have a particular density that you're looking for. And these are nice because often what you do, instead of doing fractions off the bottom, which you can certainly do, you can just stick a needle in the side and pull out that band that you're looking for. But this is exactly what was done with the N15, N14 DNA, was looking at these kinds of equilibrium gradient ultracentrifugation. So you can get very fine definition of these different densities. You know, N15 versus N14 is not a huge difference in terms of your DNA, but you can get them separated using this process. So those are your analytical centrifugation processes. Now I want to talk a little bit about chromatography. This gets back to that whole fractionation stuff that we talked about before. Here, it's actually kind of like a gel that you put something at the top, you let it go through, you see how fast it goes through, and depending on how fast it goes through, you'll be able to separate things relative to each other. Originally, this was used for separating dyes, in fact, into different colors. And so you can do chromatography separating colors, and that's, again, where the whole idea of chromatography came from in the first place. This is looking at a slightly different process, but again, if you've got a whole mixture of colors up here at the top, you now let them go through this matrix, which has something in it that's going to slow some things down more than the others. So in this case, it's slowing the green down more than it slows the red down. You end up with now fractions from the bottom here, gray, gray, gray. Then the red comes out, then the green comes out. So the real key to these kinds of processes is what's in this solid matrix. What are you using to slow down the proteins which are in here? There are basically 4 different ways. We can have ion exchange, which is just looking at the surface charge of your protein. Hydrophobic interactions, again, looking at the hydrophobic parts of your proteins. Gel filtration, which is just literally looking at size. And affinity. Now, we'll look at a couple of those here. First of these is ion exchange. On the left-hand side here, ion exchange just has something in your matrix which has a defined charge. In this case, it's going to be a positive charge. That's what's present in these gray blobs. We've got lots of positive charges there. So if you put your protein through here, and it has a negative charge, that's going to be bound to the matrix and slow down anything which has a negative charge. And it has a positive charge is going to come straight through your column. Once it's bound, how do you get it off of this positively charged part, matrix as they call it, to pull this off the column? You just increase salt concentrations. And that's where you have your exchange, your ion exchange, where the salts will bind to the positive charge and your protein will actually be released. Another one of these matrices is again called gel filtration or size exclusion chromatography. Now instead of having your matrix with a charge on it, your matrix has holes in it, holes of very defined sizes. If you have a small molecule, it will go through these holes. If you have a big molecule, it will not go through these holes, so it's an exclusion. So the red ones are getting retained, the green ones are going through. These two methods are really nice to do early on in your purification when you've got a whole mixture of different proteins, because you can put a lot of material, particularly on an ion exchange column. The problem is it doesn't separate these proteins very well from all of the other proteins. And again, we'll just use the example of the specific DNA binding protein inside cells, which are very low concentrations, but you still are very interested in them because they're probably important for regulatory purposes. So these will give you about what we call a 20-fold purification. So if you start out with 1% of the protein you're interested in at the beginning of this experiment, after this you'll have 20% of the protein that you're interested in. On the other hand, we already talked about affinity chromatography. This is the way of pulling that needle out of a haystack. If you happen to have a very specific way of interacting with just the protein that you're interested in, of all the other proteins which are present in your mixture, for instance, a DNA binding protein that you have a specific sequence for, now what you do is you attach that very specific thing to your matrix, so these black bars right here. Now the only thing which will interact with these are those that have a very specific interaction. Everything else goes through your column, and you end up with very high amounts of purification here. Now, thousands of fold of purification and sometimes even higher. So we already talked about DNA. Another very common way that people use these, and have an antibody. You have an antibody to your favorite protein. You put that antibody onto the matrix. You can just purify whatever's going to interact with that antibody. You can have, if you have a protein that you know binds to a particular ligand, say a conatase that binds to iron, you put iron on your matrix. And so you run everything through it, you'll find all the iron-binding proteins that associate with that. And these days, what a lot of people do is they put onto their favorite protein, with techniques that we'll talk about on Wednesday, what we call a tag. And basically, it's a, hi, I'm the protein you're interested in. And you can use me to pull the protein that has that tag on it away from all of the other proteins that you have in the cell, or the vast majority of them. His tags, which are multiple histidine residues, people use a lot. People also use these, what they call flag tag. Flag tags are just literally a stretch of amino acids that there are antibodies that will react with. And people can buy, you can buy those specific antibodies. You can also get proteins that bind to specific ligands, like the maltose binding protein or a glutathione binding protein. So to finish up, let's just talk about a couple of examples here of this kind of purification. So what we're skipping here is the centrifugation. So we have our cells, we break it into smaller pieces, and then we find, particularly say again, we'll take our specific DNA binding protein here, that's going to be in the nucleus. So in our nucleus, we will try and separate out our particular protein. And this gets back to one of your questions about how do you know which of the fractions has the thing that you're interested in. So in this particular case, again, we have a DNA binding protein. You'll look at every one of those fractions and see which one of those fractions has this activity that binds to your particular DNA, like a band shift assay, an electrophoretic mobility shift assay, for instance. So you have your extract, and then you put it on one of these ion exchange columns. You'll see lots of proteins that come through here. These are the ones which have the same charge as whatever you've put in. And then you increase the salt, and here this red line just represents the total protein which you have in there, and then you'll see this little blip here. That's where you have your activity. These are these individual fractions. So you put these fractions together. Again, you found their activity, say binding to a specific DNA by electrophoretic mobility shift assay, and put them onto a gel filtration, a size exclusion column. Here again, you'll have individual peaks, which are now still mixtures of protein, and a few of those fractions have your activity in them. And so here we have that second step. And then the third step, which would be your affinity chromatography. Here you have the vast majority of proteins, and I just crossed them off here because this should be way, way, way up high here. And then the only thing which is binding is this protein that you're particularly interested in. So I promised some real STS page shells. And so there's this one here. These are on the outside, as we were mentioning before, your ladder, or this is the relative molecular mass. You start out here with a mixture of all of these different proteins. This is actually literally what you'd put on one of your columns. And then after that, this blue stuff here turns out to be 3 different proteins. And then finally, 1 protein after a 3rd column. These are actually both affinity columns. This is what you start with. You end up with eventually this protein. And you'll see here there was very little of these proteins at all when you started with them. And eventually, you're able to purify these away from each other. Any more questions on this? What does it mean? Yes? So these are 3 separate purifications. So here, our cell extract. This is, and in fact, a very clean cell extract with very few proteins that are in it. Then you have in a column which has cyclin B2 that's bound to it. Cyclin B2 binds to these 3 proteins. And of these 3 proteins, only one of them binds to cyclin B3. So there are different columns with different, in this case, different affinity things that are going to bind to them. Yes? Delay the clicker question. Go for it. Yeah. Yeah, so why do it in this order? Your ion exchange to gel filtration. So the main reason you're going to do that is if you remember the way that these things work, they've got a charge, and so you have a charge-charge interaction. That actually can concentrate the protein that you're interested in, because it's actually bound to the column. The gel filtration column is actually diluting your protein because there's nothing which is specifically binding it to your column. So that's why you do the ion exchange first and the size exclusion second. Okay, we have 3 minutes left, so we've just enough time for the clicker. We do now the click, talk, think, right? Okay, so see if this actually ends up working. You're trying to purify a general transcription factor. Which of the following techniques is likely to be successful in giving you the purest protein? We actually get this thing to start. That would be nice. Go. And if we get 100% the first time, everybody can leave early. Okay, we're talking already. We're obviously not thinking anymore. So let's stop this and talk really quickly. Yes? What do we think's right? Start it again. Okay. Voting again. Do it again. We weren't at 100%. I still want 100%. Come on, let's go, guys. Ten. 3, 2, 1. There's still people who are messing with me. There's still people who are messing with me. I don't know who you are. I probably could figure out who you are, but I'm not going to do that. So yes, it is affinity chromatography. We'll see you on Wednesday.